All right, so I think we're going to go ahead and get started. We'll probably have some tricklers from the plenary upstairs, but it's great to see everybody in person here today and a few people online. My name is Darcy Dugan, and I'm the director of the Alaska Ocean Acidification Network, and I'm with the Alaska Ocean Observing System. The Alaska Ocean Acidification Network started in 2016, and our mission is to work with researchers and stakeholders to try to advance the understanding of ocean acidification processes and consequences in Alaska. And tonight is an opportunity to hear from the ocean acidification science community about the activities they've been up to, ranging from open ocean monitoring to nearshore to lab-based studies, and to get updates on that. This field has really grown in the past 10 years. We now have about more than 25 people that are involved in ocean acidification monitoring in some way, whether it's the focus of their research or their mooring. They agreed to put a weigh sensor on it. And if you count the community samplers around the state, that number is almost doubled. So we have 9 speakers today. They're each going to provide a 5-minute update. And I'm going to have a little timer on my clock that's going to my friend the duck is going to help me know when it's time to move to the next speaker, and we'll hopefully have time for 1 or 2 questions from each of the updates, and the speakers can be answering questions while I'm queuing up the next presentation. And then we have this room until 7, so I hope that what we hear today spawns more ideas for collaboration and working together, and then I think a lot of us will be migrating over to the posters to hopefully continue those conversations. So we have a few people online. We have 8 presenters in the room and 1 person, Lauren Bell, who will be presenting from Sitka at the end of our lineup. So without further ado, I think we're going to launch here with Jessica Cross from NOAA, and she's going to be talking about open ocean monitoring and upcoming summer cruise. And let me see.
So while Darcy brings up my slides, I'll just get started talking to everyone. Hello, my name is Jessica Cross. I work with NOAA, as Darcy said. I'm based out of Seattle, but almost 100% of my portfolio takes place in coastal Alaska. I went to graduate school here, and it is so exciting to actually be back at AMSS when there's actually solid snow on the ground. Really happy to see the cold snap hit you guys and for it to still be sticking around while I'm here. As part of NOAA's program, I work with a group that's called the Alaska Enterprise. We're an interdisciplinary group of researchers that studies ocean acidification from end to end. So all of the way from can we observe ocean acidification to what are its socioeconomic impacts going to be on the other side? What is the relevance of the observations that we make for the biology, for the ecosystem in these places, and then for the human communities that depend on those ecosystems for cultural and food security on top of that. Tom Hurst, who will speak later, is our fearless leader. This is sort of our portfolio from 2017 to 2019. We were able to make most of the measurements that you see here in this plot, including imprints in the Gulf of Alaska, the Bering Sea, and the Chukchi Sea. We coordinate pretty extensively with a wide variety of partners through the Ocean Acidification Research Center at the University of Alaska Fairbanks, including government, state government, and private industry and tribal partners as well. In 2020, we have some upcoming field missions that will focus on ocean acidification. One of them is an OA intensive cruise that will take place in Southeast Alaska. One of them is in conjunction with the NMFS stock assessment survey in the Bering Sea. We're really excited to have this newly funded project take place in the Bering Sea this year. It'll be the first time in 10 years that we've been back to the Bering to collect discrete ocean acidification measurements from a ship. And we're hoping to be able to turn that into an OA index that we'll publish as part of the ecosystem status report. I realize that was a lot of jargon. If you're interested in how OA is going to feed into the management community in the Bering Sea, please ask me afterwards. I don't have time to explain everything in 5 minutes. We'll also this year be heading up into the Beaufort Sea, which is something that we've never done before as part of NOAA. And I'm really excited to be participating in sort of the Beaufort extension of the Distributed Biological Observatory. Overall, what we care about is the fact that ocean acidification could threaten these commercial and cultural assets around Alaska. So in addition to that monitoring portfolio, we've also started trying to work with a number of small communities on adaptation strategies. This is work that is just getting off the ground. We're still sort of figuring out how all of this is going to take place. But if you are a community leader or a boundary organization in the room and you have an interest in talking sort of climate solutions and strategies for your community, I would also be interested in talking to you about that. I will take questions now, I think. How'd I do on time?
Awesome.
Great.
Any questions for Jessica?
Stunned the room. Love to see it. I don't know, I think Wiley is going to be up next to talk about more of the near shore rather than the open ocean perspective.
Yes. So Wiley Evans from the Hakai Institute.
Thanks, Jessica. So yes, Wiley Evans from the Hakai Institute. I am the project lead for a project we have that involves monitoring CO2 parameters from the Alaska ferry. This is a co-funded project with AUSE, the Tula Foundation, and ACRC, the Alaska Coastal Rainforest Center. We've been doing this project for 2 years now, and I'm just going to show you a bunch of data for the remaining 4.5 minutes. But basically, what this ship is, this is the Columbia, so this is the flagship of the Alaska ferry fleet. It goes from Bellingham up to Southeast and back every week. And so every 2 minutes along that weekly transit, it's making measurements. So far, we've collected just under 300,000 measurements in the last 2 years. If you take this map and you sort of rotate it a little bit and you look through time, that's what this plot on the bottom is showing. So this is the measurements from the ship. Each line is one transit, and the color is the CO2 content. And so what we're seeing here is the first year and the second year of data. This is winter into summer, and you can see there's big spatial gradients along this 1,600 kilometer transit, and there's a lot of temporal patterns happening. So there's a big seasonal cycle. The blue is CO2 being drawn down by phytoplankton during the spring and summer blooms, and then it returns to yellow, high CO2 in the winter. So the winter conditions are very high CO2. The summer, in areas it can get drawn down because of the blooms. You can see regions where it maintains high CO2. These are mixing zones. And so it's a really complicated mosaic of time and space patterns. And we're now beginning to see year-to-year differences as well. And this shows up a little bit better in this second image. This is aragonite saturation state. So I don't think Jess explained or mentioned what that term is, but essentially this is a term that tells us how favorable the water is for building shell material. And so it's a ratio, and when it's less than 1, it's not very favorable for building shell. And so you can see in the winter times, it's the same plot, it's red or to this white, meaning it's really not favorable everywhere. And then during the blooms, the phytoplankton drawdown and the CO2 make the conditions better, right? And so, and you can see that we're getting higher, warmer colors during the summer, and you can start to see year-to-year differences in those patterns as well. So we have large spatial gradients, we have interannual variability, there's large seasonal variability, and then there's even within season variability. So here in the summer period, in the northern end, you can see there's a large change from blue and yellow to corrosive conditions just within the summer. That's melt. And so we're starting to see different drivers as well along this pattern. So this is an excellent data set. What we can also do with it is estimate what the change has been since the pre-industrial. And that's what these plots on the bottom show. So this is the change in pH. Very large change is red to yellow. Blue is less of a change. Actually, a global average is right about this color blue. And so you can see over most of the transit, and so each line now is a month of the year. It's more than blue, so it's a yellow to red. So we're above the global average change in pH most of the time in this domain. This is the change in, sorry, that was saturation state. This is the change in pH. So we're above the change in pH over most of the transit. The change in saturation state is actually greater in the summertime. So you see these deeper reds. What's cool here is that, not cool, but what's interesting is that you can actually subtract this from this pattern up here, and all this undersaturation in the winter goes away. And so that's the anthropogenic CO2 signal that we're reporting on now. And so this is interesting. What I want to end with is how this all fits together. This idea that Jess mentioned a couple times, of portfolio of observing. How do all these assets fit together? And in Southeast, there's a bunch of things going on that are really great to tie to the ferry work. So this is the ferry track. The color is the severity of the saturation state, which is, we've defined here as the lower fifth percentile. So the 5th percent, the lowest measurements. And these are the number of observations from a series of high resolution monitoring sites. This is the Sitka percolator. This is the NOAA mooring that was in place for a number of years until 2016. This is the Oceans Alaska percolator. And then we have a number of systems down in BC as well. And so this is the number of observations from these sites, with the black line being the severity. And so you can see most severe conditions from these time series sites is pretty close to 1, but the ferry paints a much more complicated spatial pattern. But at these sites, we're resolving the CO2 chemistry more often than twice a week. And so it's the combination of these assets that we really need to get a handle on OA in this domain. And so I'll just end with saying that Columbia is going to continue in 2020. We're happy about that, and hopefully longer. So thank you very much.
Any questions for Wiley?
Yes,
So Wiley, does the ferry just measure surface water then?
Yes.
So, and how does it vary then when you're looking at deeper water subsurface?
We see deep water signature in those mixing zones, which is why it's much higher CO2 content than, say, typical winter surface water. But yes, it's all water from 2 meters depth drawn through the vessel.
Okay, real quick, how's the sound in the back? We have a microphone that could be used, don't need it. Okay, and then there's a few seats in front. If you have an empty seat next to you, could you raise your hand? And then there might be, if you guys want to move up and sit down, feel free. All right, great. Our next speaker is Darren Pilcher. He's from University of Washington's. Could you, I always forget the JSOW acronym. Could you explain?
Joint Institute for the Study of the Atmosphere and Ocean.
Excellent, thank you. And he's
But I sit right across the hall from Jessica, Noah, PMEL. So.
Many affiliations.
Yeah.
Okay.
Okay, thanks, Darcy. Hi, everyone. So yeah, I'm an ocean biogeochemical modeler. I handle most of the modeling side of the Alaska OA enterprise that Jessica introduced earlier. But I work with a team of others at JSAO. We design these models to have the physics, the biology, the chemistry. It all gets combined so that we can have this biogeochemical model with carbonate chemistry in it. And our long-term goals are to use this to project OA over the 21st century, and then also to move more into seasonal prediction capability, so 9-month forecasts. I'm being told to talk louder. So for this little session, I'm just going to talk about dynamical downscaling. It's a process we used for these OA long-term projections. It's essentially where you take a global climate model and use the atmosphere and the boundary conditions that it has, like the water temperature coming in from the sides of our model, to force our smaller regional model forward in time. The advantage of doing this is that you get higher spatial resolution in our regional model, and also a lot of the coastal biogeochemistry, like freshwater runoff that Wiley was just describing, that isn't in a global model, right? Our global models are used more for capturing large-scale ocean basins. At this time, they're not really designed to be zoomed in on for a particular coastal shelf. And so for this, we ran 6 total simulations using 3 different Earth system models, 2 different emission scenarios. So one is RCP 8.5. That's the very high end, business as usual, burn all the carbon everywhere sort of emissions. And then also RCP 4.5, which follows 8.5 closely to about 2050, but then levels off and eventually emissions decline by the end of the century. So this is what it looks like when we have all 6 simulations thrown on a plot together. This is a time series of surface aragonite saturation state. For each model, the red lines are the MyROC model, blue is the GFDL model, and black is the NCAR CESM model. And the point here is to show that there is variability amongst them. So for each one of these, we're using still our regional model, but it's just being forced by the atmosphere from a different global climate model. So there are large differences in those climate models with surface temperature forcing, with the amount of carbon and alkalinity that are coming in. And so that manifests into changes for our projections of ocean acidification over this time frame. But just to point out that both CESM and GFDL tend to converge right around the value of 1 on an annual time frame for surface omega by the end of the century. Bottom is on the right. There's a bit more scatter there, but overall it's much lower to start with. We pass that value a lot quicker. This is a very spaghetti kind of confusing diagram, so we can also coalesce this into just the mean between all 3 models for each scenario. So showing more of a best guess estimate of what the conditions will look like over the coming 80 years. And again, showing that in 8.5, we get close to that value of 1, as Wiley was describing earlier, by the end of the century. 4.5 follows 8.5 closely for a while, but then kind of levels off as our emissions level off. The bottom is a little bit different, and then that threshold is crossed much quicker, 2030 for RCP 8.5 and 2050 for 4.5. But as Wiley mentioned, there's a lot of seasonality in this, so maybe looking at the annual average isn't the best picture. Here is just a graph to sort of summarize the seasonal cycle. This is in 2020, showing at the top the seasonal max, so how high omega gets because of biological productivity. The lows at the bottom, the min during that winter time period, and then the mean. It shifts to this by 2050, so even all 3 move downward, but the min not as much as the max. So we get a little bit of a compression of the seasonal cycle. And then by 2100, it's compressed even more. And in fact, at this point, right, the seasonal max that we have now in 2100 is actually less than the average conditions occurring today. So just trying to put it in a little bit of context with these numbers. And that even our seasonal min in our mean in 2100 is very close to what the minimum values today are. And then I'll also just wrap up showing, of course, with these models, we also get a lot of spatial information as well. This is the change in omega over this time frame for all 3 models at the surface. Again, if we take sort of an average picture and say, where do these dominant spatial patterns emerge, we get this picture where generally the outer and middle shelves are the areas that this model is projecting to acidify relatively faster than the other areas. In particular, the southeastern Bering Sea is where a number of these models, you can see it most clearly in MyRock, but it's also in GFDL as well, that sort of emerges as one of the hot spots for these projections moving forward.
Questions for Darren? Should make a plug that Darren is speaking upstairs from the plenary session tomorrow morning at 8 a.m.
I was going to mention that, yeah.
He will be able to go
into
more detail, yes. Thank you. So our next speaker is Andy Wall, and she's with the Kodiak Area Native Association and is going to be talking about the community sampling program that started up this past year.
Like Darcy said, my name is Andy Wall. I'm one of the environmental coordinators at the Kodiak Area Native Association, also known as CANA. CANA is primarily a healthcare facility, but 2 years ago, we got funding through the EPA IGAP program, consortia funds. And so we received those funds about 2 years ago and were able to start this monitoring program. I'm still pretty new to the program, probably about 2 months into it, so please forgive me if I misspeak. But the previous environmental coordinator, when we received the funds, went to all the tribes on Kodiak. There are 10 federally recognized tribes on the island, and CANA serves all of those communities. And so she went to the communities and identified the needs, and water quality monitoring was one of the major concerns of tribal members. And so she reached out to Bob Foy, who was the NOAA lab manager, the Kodiak lab manager, and he was already doing ocean acidification work. And so that relationship or that network kind of happened organically. And so the program, they discussed the current efforts that were happening and then how the tribes could fit into those efforts. And both entities knew that Sitka and Chugach had started a community-based monitoring program and kind of used that as a model to build our own program. And so the program itself is really kind of baseline monitoring. We sample the IGAP coordinators at each of the villages, samples once a week at the respective locations, and measures for pH, dissolved oxygen, carbon dioxide, temperatures, and salinity. We use a YSI and a Niskin, collecting at the surface and then the bottom. So both of those samples are fixed in a beer bottle with mercuric chloride and shipped to the NOAA lab, where there is a percolator tender, Carrie Mason, who analyzes those samples and reads those results. And so all of these locations have IGAP programs. And so it's within this last year that the program started. So these are the red stars that you see are the current monitoring locations that we have. One at the South Trident Basin float dock, which actually the percolator tender collects that sample in addition to other NOAA people, I think. Mike can correct me on that. I think he's speaking next. And then there's also one at Larsen Bay, Old Harbor, and Uzinki. Port Lions is that upper yellow star that you see up there. They have new environmental coordinators, and so they need to receive training on how to collect these samples. And then Akyak down on the bottom does not have an IGAP program, but has a really dedicated volunteer that wants to start sampling. And so we're able to help facilitate that location startup. And there's more sample locations on Kodiak that maybe Mike can touch on, but these are the ones that CANA is directly involved with. CANA coordinates the coordinators, and NOAA does a lot of the supply coordination. We also host a marine water quality workshop last year. This is how the environmental coordinators received the training to conduct the samples. And so it was a 2-day training, heavy intensive on education, and then hands-on sampling of how to actually collect them. And then this last year we did the same thing and are actually making our way to each of the locations to ensure that the samples, the accuracy of the samples are being collected. That's the current program. Any questions? Thanks.
Where's the data go?
So the data goes right now to, Carrie Mason is collecting it. And so this last year with the change, the environmental coordinator that left, the percolator tender left, and then the lab manager left. So there was kind of a lapse in data for this last year. So samples were very spotty, but all of those we have and are holding on to. And then our kind of within that year time frame, we're going to make a plan of what that looks like. So we might be reaching out to you or to, I'm not really sure exactly what the plan was for that, but definitely coming together with the tribes and NOAA to figure out how to interpret that data and how to distribute it.
Great. And as Andy mentioned, the Kodiak program is joining programs, or I guess an additional networking site now in addition to the Sitka Tribe of Alaska that runs a sampling program with 7 or 8 communities in Southeast Alaska, and then the Alutiiq Pride Shellfish Hatchery, which is, there's another 7 or 8 communities that are sending samples to them as well. So we now have just over 20 communities across the state that are doing weekly water samples. Our next speaker is Mike Litzo. He's the director of the NOAA's Alaska Fisheries Science Center Kodiak Laboratory.
Thanks, Darcy. Yeah, so as Darcy said, I'm the new lab director at the Kodiak facility, NOAA facility. I'm also the program manager for the Shellfish Assessment Program for the Alaska Fisheries Science Center. I've been in that job since November, and so I'm taking over for this really good program, but all the work I'm going to be talking about is work that's been done by others. So we have a Berkelator on site, and we are cooperating with CANA in the program that Andy just described. And then in addition to that, our group's been involved in research on acidification effects on commercially important crab species in Alaska. There's been a number of studies that have gone on over the past decade focusing on these 5 commercially important taxa. And the group's gone through in a really, what I think is a pretty admirable way in focusing on different life history stages and different mechanisms where you might see a response to acidification. Those are outlined here. And the color coding is helpful in sort of interpreting this, where red indicates a negative response that's been demonstrated in the lab, yellow indicates a neutral response, and green indicates a positive response. So that's sort of the one slide summary and what we've done so far. And as we've gone through, or as the group has gone through the different taxa and the different life history stages and the different mechanisms in this fairly systematic way, it starts to give you sort of a broad enough understanding that you can see some surprising results start to appear. So this is a good example of that. These are sort of hot off the press results having to do with fecundity effects on snow crab of ambient, comparing ambient conditions to 2 acidification treatments. Take home is that there appears to not be an effect in our laboratory studies. And that contrasts really strongly with a congenitor, tanner crab, where in the same experimental conditions, you see a really strong reduction in fecundity. So the sort of breadth of experiments that have gone over the last 10 years are giving us this ability to make some comparisons among taxa. In terms of how this program's fitting in with a sort of a holistic view on how commercially important crabs are going to be affected by acidification in Alaska, we've mostly studied direct, or almost entirely studied direct acidification effects to date. And we've got a lot of interest in our program in looking at interactive effects with other stressors like temperature change. And then we're aware that there's a whole range of indirect effects with things like change in physiology, the change, say, feeding ecology of animals. And those, we recognize as sort of further goals that we can start to address in the program that haven't been addressed to date. Chris Long, I should say, is our lead for all this work, and he gave me this outline for sort of the next steps of where we see this program going. And I won't read that to you. I'll let you read it yourself. But I do want to take this chance to put in a plug for potential collaborations. So this is a really cool group of researchers in Kodiak that's put together quite a good program over the last 10 years. We've got a great facility. And they've worked out the animal husbandry issues, and they've worked out the experimental, the mechanics of running these experiments successfully. But we're also a small group. We've got a lot of responsibilities for assessments in the Bering Sea, running the trawl survey. And we've got responsibilities for other research on crabs in the Bering Sea. So we're really limited right now by people power. We've got a lot of lab capacity. So if anyone out there is interested in collaborations with our group, I'd love to hear from you. Thanks. Any questions?
All right. So thank you, Mike. Next, we've got Marina Washburn, and she's with UAF. She's part of Amanda Kelly's lab, and is going to be talking about bivalve response.
Hi, everyone. So this is some data that I've collected from a project we started in 2017 and happened in 2018. And it was based off of Amanda Kelly's sensor data from Katchmack Bay. So a great connection between all of the kind of network and modeling information you've heard and then applying it to an actual biological experiment. So we did an experiment looking at the impact of elevated and variable PCO2 on larval razor clams. And we thought the variability would be the big story. hasn't been studied a lot in experimental applications just because it's kind of hard to mimic in a lab environment. It turns out razor clams are just very strange, and so that kind of has become our story that we're following right now. They use a larval technique for shell development that's more commonly found in gastropods or snails. And so they delay the onset of calcification until much later in their early life stage. So we've been looking at that, and actually here, so what you're looking at is overall shell composition over time. On the far left, we have day 7 all the way through day 28. So day 7 is kind of the first time they really have any kind of shell structure. And then we examined 2 points per shell, and those 2 points are represented in the far left bottom corner here. And essentially what we see is that there's a large amount of contributors to overall shell composition early on, which is consistent with that unique developmental strategy. But later on, we do start to see calcification beginning, and this is indicative not only in visual samples, but in this composition. So we see that calcium is starting to become the majority stakeholder in terms of composition. And actually, interestingly, is occurring first in the high exposed treatment and then in high and variable. So to us appears to be some kind of indication of developmental stress due to the elevated PCO2 conditions that they've been exposed to. Yes, so the other thing was looking at the type of calcium carbonate that these shells have been using, because there's quite a few different kinds and they're not all created equal. And so what we found was that early on, these organisms, because even though it's not a traditional shell, there is still calcium present. Early on, they're using amorphous calcium carbonate, which is the most soluble or sensitive version of calcium carbonate. And then later on, we see veterate, which is very odd because veterate is not very biologically understood. We know we find it in one larval snail, a freshwater snail, and then in sea turtle eggshells. So not very well understood, but it's found in our razor clams. And again, unfortunately, this is another highly soluble form of calcium carbonate. So this was really confusing, but it turns out when we look at just geochemistry, so not a biological side, we see that sulfate to carbonate ratios play a really large role in determining whether or not veterite or calcite is formed. And what does this have to do with OA? If we look at how ion concentrations are expected to behave over time, there should be less carbonate availability, meaning that that ratio would favor veterite over calcite formation. And we do see this, the pattern of this indicative in our sulfur levels in our high and variable treatments, so those that have been exposed to elevated PCO2. So I have a poster on this tonight. Feel free to come and say hi. I also have one we did an experiment last summer on the black chitin, or the badarki, or the Katy chitin, many different names. There's some preliminary results from that. And we'll be doing the first OA salmon experimental study this coming summer on juvenile pink salmon. So taking all the lovely sensor data and applying it to animals that are important to Alaskans. Thank you. Oh, yes. Sorry. Thank you. Yes.
Of your mineral differences that you listed there, veterite and calcite, what is the mineralogical difference between the two?
So veterite is more soluble than calcite. Calcite is, I mean, if you can say the best form of calcium carbonate, it's the least soluble. It's still prone to dissolution, but not as much so as veterite. ACC is barely a crystal. It's very, very loose. And veterite is kind of just like larger ACC. So not great in terms of sensitivity.
Do you know how veterite compares to aragonite or calcite?
So yes, it goes ACC is the worst, and then veterite, and then aragonite, and then calcite. And high mag calcite, some people say it's worse than aragonite, some people say it's better. It depends on what study. I think the jury's kind of still out on that in terms of biological processes, just because the individual can play a large role in determining. So it's variable. Unfortunately, it's not a straight answer. Again, I have posters tonight, so come if you have more questions, let me know.
And Marina was at the Alaska Shellfish Growers Association meeting in Ketchikan last week.
Oh, yeah?
keeping them up to date on these recent.
Oh, yeah, the long range shot, eh?
So next is Tom Hurst. Tom is with NOAA's Alaska Fisheries Science Center based in Newport, Oregon, with a focus on groundfish response.
If it didn't involve jail time, I'd go 100.
Thanks, Darcy. Thanks, everybody, for coming. I'm just going to point out that this is work done in the laboratory on studying the groundfish, the effects of OA on groundfish. I'm going to spend a little time just to sort of orient you to the kind of work that we're doing, and then I'm happy to talk for many hours about the specifics of one of those studies. But the kinds of the approach that we're studying in the fishes, there's the kind of effects that we've talked about in the other critters that we just heard about, like these sort of direct effects that the high CO2 cause in terms of growth or survival or fecundity or in the bivalve shell thickness and that kind of thing. And then Mike mentioned this a little bit, the food web effects, the idea that OA is going to affect the food of fishes. We have to keep that in consideration when we think about the cumulative effects of OA on fishes in the future. But we also know that in fishes, there's an effect which we probably don't see in some of the other animals, which is that work has shown that ocean acidification has the potential to disrupt some of the sensory and behavioral biology of fishes. And fishes have a lot more broader repertoire than other animals. And so that's an aspect that's a little bit, so far, unique to the work on fishes that isn't seen in the other organisms. And then the idea is by looking at a range of these different kinds of effects in different kinds of groundfish species in Alaska, we'll get a really broad picture of basically what's going to happen, and then try to pull together all of that into sort of summarize the cumulative effects over time of OA. So I'm just going to very briefly point out a couple of things that we've done over time. And again, I'm happy to talk in much more detail about any of these things. So just some of the direct effect studies that we've done so far. We've done studies on pollock, cod, and northern rock sole. One good thing we've seen is that the walleye pollock, which was the first species we studied, seems to have a pretty strong resilience compared to some other fishes too. Although I will point out we have one of my colleagues, Michelle Stoll, is up in the post session following up on that and some of the
putting stuff on the pizza.
lipid metabolism related to ocean acidification. So anyways, you may want to check that out for some news that's maybe not perfect for walleye pollock. But we have seen some sensitivity in larval northern rock sole and larval Pacific cod. And so again, we're not as far down the path with studying as many parameters and as many species as they have in the crabs, but we're working our way there with the groundfishes.
Your balloon popped?
We have been doing some of this behavioral disruption work, and just sort of I'll just sort of tell you what this is. In one study, basically we found that juvenile pollock are less likely to recognize the scent of their food when they're exposed to high CO2 conditions. And so it's a very complicated experiment, but just to say that's what's shown in the panel on the left. The panel on the right shows that larval Pacific cod change their behavior in a light gradient, so how they move around in the environment when they're larvae and how that is going to affect their ability to locate food catches is the kind of things we're looking at in there. Sort of following up in some of the more nitty-gritty about exactly how those kinds of behavior sensory changes are caused. I've been working with a colleague at Hampton University who's actually looking at the transmission of nerve signals in the brain, in this case related to hearing, but trying to really understand, really what is it about, what the biological, the neurobiological underpinning of those kinds of effects that we see in the kind of behaviors. And then a newer project, some of the both behavior and growth rate effects that we've seen in Pacific cod. Now we're trying to sort of put those things together to try to predict what the spatial effects of potentially OA impacts on Pacific of the stuff that we know, plus using the model that Darren presented earlier on. So merging the field observations, the modeling, and some of our laboratory experiments sort of come up with this long-range prediction about what the possible consequences of OA are for, in this case, Pacific cod. I think that's it.
Any questions for Tom? And as Tom mentioned, he's got a poster at the poster session
Beautiful. You did a very good
Okay,
job.
our next speaker will be Dave Finhoff from
Can I try
University
another
of
color?
Wyoming.
Can I try
And
color?
Dave is the
Oh,
head
I
PI
considered
on
it.
a study
I
looking
see.
at the response of salmon to
Start
ocean
over again?
acidification and environmental tipping points.
Okay, maybe I can try the brush.
And actually, there was one request about muting
Mm-hmm.
our remote speakers. Oops, sorry, I advanced you by accident.
Thanks. Thanks. I'm glad to be here. I'm an economist at the University of Wyoming, so I'm a little bit out of place. Reason I'm interested in this project is that I really am interested in helping to inform policy making before risky events occur. So how do you behave in the intervals of time before something might change the way that people operate? And from listening to you guys and listening to a lot of other people about this, it appears that we could be facing a regime change from multiple stressors. And how can we develop policy that helps inform that? This is also joint work with Joao Vaz, but also a whole bunch of people that, including some people in this room. As you guys know, there's little is known about of this until work like what Tom just talked about and what Amanda and Marina have been talking about. But essentially, we're at the start of a research program. I was down in Miami listening to a bunch of talks about ocean acidification in general, and it seems like overall the geochemistry is starting to be understood after about 15 years of really intense work, and the implications now I think are people are starting to dig into it. And what we want to do here is take a prime case here where we've got a really important situation and say, hey, can we help inform policymaking early on? We're going to just look at pink salmon as a case study, but we do think that like the kind of principles that we're trying to start here in the big integrated project, hopefully would be able to be translated to other key species of interest. Okay. The debate when you bring an economist to the table, they're going to really start talking about marginal this, marginal that, right? And you guys are probably sick to death of people like me telling you that you need to change your lives or how you operate. But what the models always boil down to is that we're really trying to say, hey, what's the marginal cost of essentially letting one more unit of stock escape today versus what do you get in the future of this? And this is, you know, in terms of those of you who are familiar with stock assessment, something very similar, except you're not essentially thinking about optimizing. You're thinking in the way that we might. You're thinking a little bit more about MSY, but it's a very similar type problem. What we really see in the classic case, this would be for an escapement model with thinking about spawners and recruits and an optimal escapement level and a carrying capacity type level, an equilibrium type measure, is that in terms of the economic policy insight with this typical kind of Rickert type function here, is we have essentially an interval of if your stock is less than this level, we should conserve. If we're at this kind of target level, we want to sustain that into the future. And if we're lucky enough to have stock above it, we want to we can essentially aggressively harvest us back to this kind of level where we sustain. And this is how we might think of the world now. But what we're interested in is what happens if, and this is a hypothetical case, and where we're trying to blend the work of people like Tom and Amanda and Marina to say, what if this combination of multiple stressors changes from Rickert type principles to something more like critical depensation, where really you have the emergence of a threshold in the population where essentially if we have stocks less than this, we're going to drive them to extinction. Well, as an economist, what you want to tell people about is, well, the economic models in this setting, they're really drastic. They basically say if you have a stock less than that threshold value, then essentially your asset's going to disappear, so you want to liquidate. So the economic principles or the incentives, and people react to economic incentives, are to liquidate the asset that's going to be gone before you lose it. So they use it or lose it. If you have this kind of an effect that comes in through the combination of stressors, then you bring up this kind of potential for a liquidation of that asset. And it means that I call it a window of conservation, but the stocks that you should basically do nothing and let the stock
kind of grow up or expand to the level where you've got a constant escapement, it's narrowed. So there's some real kind of implications here on whether or not we're kind of, by this regime shift, put into a use it or lose it situation, or if we're in a precautionary management type situation. And what we want to see is there potential for this, and what makes this more likely to occur and what makes it less likely to occur. What we're really interested in here is if you're currently, let's say, managing according to the blue line, but you're facing a potential line, red line here, let's say in the future, if you were here and we have this regime shift at time tau, right? It's really bad because then we just liquidate your asset essentially. If, however, you take steps back in time and you're able to take this risky change into your decision-making process earlier, essentially it gives us a motivation to conserve. So the question is, if we do see changes that might result in the emergence of these kind of critical thresholds, let's say in the ecology, then as long as we take action early enough, it might be optimal to actually conserve and get ourselves to a new, let's say, optimal escapement out here. If we stick our head into the sand and ignore it, we could be facing a sense where the regime shift occurs and our current escapement goals have made our asset immediately in this kind of liquidate zone. I'm not saying we're going to be there, right? But it's what we want to then inform policymakers with, because if you're going to tell people to change the way they are operating their business here, we're going to, let's say, argue for more conservation, then they've got to be able to say it's going to pay off in the future. And that, if it pays off in the future, just depends how you manage it now. If your asset now is, let's say, a better asset than it will be tomorrow, then your incentives are just to use it before you lose it. We don't know where we're at yet, right? But our goal of this integrated type work is to say, what are the chances that we are facing a situation like that? And are we early enough to make decisions so that we can preemptively, let's say, help people avoid it? Am I within my 5 minutes? Okay, good.
Questions for Dave?
Yeah.
Yes, sir.
So maybe that's what you're saying works on single species management. When you have multiple species in an ecosystem that has to balance, when you start use it or lose it type of thing, you're ignoring the fact that the ecosystem component of when you take a species out.
Oh, I completely agree. And again, it's probably more a fault of the simplistic economic model that it would treat it as a single asset, you say, as you say, instead of a portfolio. But for us, it's kind of a first principle. What's interesting to me, though, is kind of management. Like we've been, part of our stakeholders are their managers assigned to these species, and they seem to be really species specific. Groundfish is a little bit different. It's more of a portfolio. But the salmon, anyways, I agree with you. And I'd say that the model's probably a little bit simplistic on that viewpoint. We view it basically as a starting point. But in reality, a lot of managers are asked to make decisions in isolation of other, in fact, wild stocks versus hatchery stocks, quite frankly. And so it's an abstraction that may not be perfect, but it might also be reflective, at least, of some of the constraints that that people face. I do think, though, in reality, most people also switch their behavior on what they target, and they move to other species as well. And we're trying to take into account some of that. Yes,
how do you recognize when you're at one of those decision-making points?
When we're at one of those, well, that's what we're hopefully going to find out. I think the question that we're going to really focus on here is trying to inform our recruitment functions here as well as possible to come up with what would these escapement rules be, if you will, and then comparing our states versus those. What we don't know at all is actually, so I abstracted a lot from this. It's a simple model, but there's actually some complicated mathematics that go behind it. But a lot of it depends upon the time of the switch, right? And if that time of the switch is way, way, way into the future, right, we've got And we just did these simple kind of what happens if you had one period back in time, how would that change your decision rule? How would it change if you have three? If that switch is very far into the future, right? So where is that tipping point? Then your chances of making good decisions now are really high. It's just if we're at the threshold. Now we're working with a bunch of people who work with a lot of you guys who are trying to see, are there thresholds in the data to tell us whether or not we're close or not, but whether or not they're able to find, I'm looking at Darcy, she's part of this team, whether or not they're able to tell us any information on how far out are we from that threshold, I'm not sure. What we want to do is say, how does the uncertainty in that timing, how should that be reflected in policy making? So the uncertainty, even though we don't know when it's at, like if the uncertainty is huge, should that say just continue as business as usual, or should it say you might want to act more preemptively? Well, it all depends on how things might look into the future. It's quite interesting. I had another question, or I might be out of time. If the single species, sorry, I'm sorry, I don't understand.
Yeah, if you're just looking at, say, one species and the whether or not we liquidating it or whether or not we urban, that window of like just letting it be shift at all with different.
Oh, different species? What was that?
No, just in a change in ocean acidification.
Oh, probably. Let's say, for example, that the changes in ocean acidification, I mean, that's what we're trying to test empirically, like how, what might occur or move from Tom's type work to these kind of life history type fields. If the impacts weren't as severe, and let's say this thing just was further over here to the left, then it would give you a lot larger windows of conservation for sure. So the severity, basically, of the severity of the impact would have a big implication here. And again, I wouldn't be telling people about that liquidated thing. I'd be saying that this is a warning that the economic signals are going to tell people their asset's going to be worse in the future. So the economic signal is to use it before you lose it. And so that what I'd be trying to say is it's a word of warning, like be careful. There's going to be incentives for people out there to get it before it's gone. And then you've got a potential for trouble. So I'm interested in the model tells us about the economic incentives of the situation. And can we then develop policies which might beat that, right? Or help alleviate those economic incentives that we've got out there. Very long-winded answer, sorry.
I should mention too that project has a governance component. There's someone at the Institute for Social and Economic Research that's looking at fisheries, like the flexibility in existing fisheries management to accommodate some of these changes. And there's also a big fisherman stakeholder component that's being led by Molly Mayo, who's, Molly, raise your hand. Over the next couple of years, we'll be meeting multiple times with groups of fishermen to kind of hear how they think about some of these issues and how they process them and what they would like to see in some of these scenarios. So our last presentation is from Lauren Bell, and she is with the University of Santa Cruz. She's a born and raised Alaskan who's currently working out of the Sitka Sound Science Center on herring. And we have the unique situation that Lauren can hear us, but we cannot hear her. So I am going to call her and put my phone next to the microphone, and hopefully this will work. A little workaround. Okay, and while I'm doing that, I'm going to let Lauren share her screen. Okay, Lauren, can you hear me? Talk a little more.
Can you hear me now?
Sounds good.
Okay. It says I can't share my screen until you stop sharing.
Okay, I will stop. Let's see here. Okay, try it. Give it a try now. All right.
How does that look?
It looks great.
All right, excellent. Great. Thank you. Great to join you all. My name is Lauren Bell. I'm a PhD student in the Croker Lab at UC Santa Cruz, but I live and base full time in Sitka, and I lead this lab's research program here in Southeast Alaska. So our lab is interested in global change ecology, which means that we are interested typically in looking at ocean acidification in tandem with other changing environmental variables. And our goal is really to scale our work on single species responses up to inform how species interactions might change in the future, especially across trophic levels within an ecosystem. So over the past 5 years, our lab has run a research program in Sitka Sound specifically focused within giant kelp forest ecosystems. This is a neat region in which to look at these systems because they reach their northernmost continuous range extent, pretty much at the top of Southeast Alaska. The heart of our research program is built around a number of field sites that we've established in the kelp forest of Sitka Sound. And here we maintain year-round benthic deployments of these sensor packages that we place at the lower depth limit of these kelp beds, so about 30 feet. We pair this environmental monitoring with a big community survey blitz that we run twice yearly in both summer and winter. And we're really interested in these seasonal comparisons because like most marine systems in Alaska, we see these big swings in environmental variables throughout the year. So here I'm showing pH in blue and temperature in red, but we of course see big swings in dissolved oxygen and nutrients and in light availability. So we're kind of considering this seasonal laboratory in which we can investigate how its yearly cycles naturally correspond to changes in organismal physiology and in ecosystem functioning. And I just wanted to give you this visual as well, just in terms of algal biomass and how it changes from winter to summer. It can be pretty darn striking. So we pair our fieldwork with a manipulative experimental system that we've built where we can isolate particular drivers such as pH, temperature, and light in order to simulate future ocean conditions. We've been improving upon this 24-tank system with a couple experimental iterations over the last 3 years. And at this point, we have it set up pretty darn well to be adaptable to a variety of organisms and questions that we want to ask. So a couple of examples of the work we've been doing that I wanted to share. Last spring, I raised Pacific herring roe from fertilization to hatch under a factorial combination of ocean acidification and warming. And in this experiment, I actually found that for some of the responses I measured, the impact of either stressor alone had opposing effects or opposite effects as one another. But when you combine warming and acidification like we expect to see in the future, that measured impact to larval physiology was either pretty minor or no longer significant. And I highlight this just because it indicates that the full physiological impact of multiple stressors may end up hiding beneath some counteracting effects of these different drivers if we're not looking at them in factorial combinations. In other experiments, we've expanded our experimental design in order to model a more complete response surface of an organism to a range of multiple stressor scenarios. So for example, here I'm showing you relative growth rate of some common kelp forest grazers on the y-axis at a variety of paired pH and temperature combinations. The 3D gray surface there represents the extrapolated average response across all of these conditions. And this approach really allows us to visualize what are often pretty complex and non-linear responses to multiple stressors. And this can help reveal some particularly stressful environmental combinations, such as we might see at certain times of year or in certain seasons in the future. We've also found that diet can play a major role in species response to a particular stressor. So here I'm showing you the change in mass of pinto abalone that we raised for several months at 3 different pH levels. And in this case, they were fed a mixed diet of common species of macroalgae. And you can see that as pH is lowered, they don't add as much mass, though they can still maintain positive growth at a future winter pH level. However, if we then limit these poor abalone to a single type of algae under the same conditions, we end up seeing that the abalone are actually no longer able to maintain positive growth in those future conditions. So this really implies that diet can be a pretty critical piece to species response to stress. So as we continue to explore these ideas, this year I am running experiments to test how major macroalgal species are going to respond to future conditions, both in winter and summer, not just in their growth rates, but also their nutritional quality and ability to chemically defend themselves. This is a picture of the experiment I am running right now. And the goal is then to take what we learn about the impacts to both the consumers and their food sources at different times of year and try and see what the emergent effects across the food web are going to be in different seasons. And lastly, I am interested in also exploring species interactions from a habitat perspective, which I'd really like to do by extending that initial work on Pacific herring to test whether an association with a photosynthesizing substrate can actually help mediate some of the impacts of ocean acidification. So that's what we've got going on so far. Thanks to Ken for sticking with me while I call it. I hope I can join you all there in person next year.
Great, thank you, Lauren. Any questions for Lauren? All right, Lauren, I'm going to hang up the phone. You'll still be able to hear us. I'd like to ask for a round of applause for all of our speakers today. That was great. It is not easy to cram that amount of research into 5 minutes, so great job, everybody. We like to think about ocean acidification research as putting headlights on a car. And every year the car lights get a little bit brighter. I hope that today's session sparks hopefully a lot more questions between people in this room. And there's time in this room here for a conversation afterwards. And I think a lot of us will be heading over to the poster session. There's some materials on the back table that have been put together by the Alaska Ocean Acidification Network. And the contributors to that are sitting in this room, and we're up here giving those presentations. So thank you all for that as well. We try to act as a clearinghouse for information through the network. So please check out the website. We interview researchers. We try to post interesting research results in kind of a story format. We provide links to events and other places to get the latest on ocean acidification in Alaska. You'll also see in the back much of what was presented here today, and then a few other researchers' work that are not here are in a 2019 Alaska Ocean Acidification update. So we started our first kind of state of the science update for stakeholders last year, and this year there's a new publication back there that shows what did we learn just in this past year. And this one more plug, there's 5 ocean acidification posters that are waiting for you at the Egan Center. 2 from Marina, 1 from Wiley, 1 from Tom, and I'm missing one. Oh, there's another on continuous high frequency measurements of pH, salinity, and temperature in Arctic lagoons that we didn't have anybody from that group today. But anyway, thank you all again. And what's that? Oh, and yes, tomorrow 8 a.m. Get a good night's sleep tonight, and you can hear more about modeling in the morning. So thank you.
Thank you.